All right, looks like it's 7 o'clock
That's
again,
great.
so we can get started. So we have Seppi Lin, who's an MD and the vice president of and head of the Omni early clinical development for Genentech, um, and he'll be discussing what is the best primary or best endpoint for phase two drug development in asthma. And he's coming to us from San Francisco. So thanks for doing this.
This is fantastic. I'm really happy to join. And, somebody in my group who leads the respiratory group, Dorothy Chung, used to be part of your clinical community and said, hey, yeah, I used to go to those. And I think it's great that you've had folks from my organization give talks in the past. Dave Choi, who is a colleague of mine in biomarker discovery, I think gave a talk a couple of years ago about seasonality of exacerbations and other symptoms in asthma. So thanks very much for having me. Um, I do have a few financial disclosures. I am an employee of Genentech, and my salary comes from Genentech and Roche, and I hold Roche stock as well as stock in other companies that I've been employed by. This is also the last slide that you'll see a company name attached with it. Some of the analyses you'll see on the following were done within Genentech, and some of that is unpublished. And so those unpublished data you may see as internal analysis. And that's just a key for some of the data that I'm showing today. And I think, this talk and I think my presence and uh, is really enabled by the sharing of data across academia and other sponsors. And I just want to stress that, I believe that data sharing is incredibly important in moving our field forwards. So what is my group and what do we do? GRED, we're an independent research and early development organization within the parent company. And my group, Omni, is responsible for all phase one and phase two studies in indications outside oncology. And that's pretty broad. Our acronym Omni stands for ophthalmology, metabolism, neuroscience, immunology, infectious disease, and all the respiratory work happens within that immunology space. We are a group of 33 people. Half of us are physicians, the other half are non-physician scientists. And what you may not know about what we do is we spend about 50% of our time on non-molecule work, meaning on work to enable new disease areas in terms of clinical development, and also to enable molecules that are coming to us from our research side of the organization. And how do we do that? Besides opening new disease areas, and we work on improving the way we study disease areas that we're already in. And this could be earlier proof activity or demonstrating new clinical relevance within those established disease areas for us. And a big part of how we do this is through endpoint development. Key innovation of developing novel endpoints. There are a few different buckets that we do this in. Right now, a key theme for us is working on imaging, and this is across disease areas in the group. We believe imaging is important in a prognostic application in terms of segmenting patient populations, enriching for populations that progress more quickly, for example. And we believe imaging can be a sensitive and early efficacy outcome also in diseases where we sometimes have to do 2 to 3 year clinical trials to get our first efficacy readout. And since we're an early development group, pharmacodynamic markers are very important for us to get an early understanding of whether a molecule is active and at which dose range those molecules are active in. A nice example of this that we published relatively recently, we have a TRPA1 program. TRPA1 is a uh, calcium channel on neurons, and those neurons are involved in neuroinflammation potentially, and also on airway smooth muscle. It turns out that mustard oil, or AITC, is a potent agonist of the TRPA1 channel, and we can actually apply AITC to the skin and through laser speckled imaging, determine the amount of erythema that is present and how much of that erythema we can block with a TRPA1 antagonist. We do this in real time. And so within 3 days, we know the PK or exposure of the drug and how much we're modulating the erythema as part of this skin challenge. that was instrumental in that program in helping us define the active dose range. But more importantly, and probably what you're more interested in, is around the clinical efficacy side. We do a lot of work with composite endpoints. That was true for our work with inflammatory bowel disease. And we do a lot of work in PROs. Most recently, in collaboration with the CPATH consortium, we used one of our phase 2B studies to qualify the ADSD and ANSD, that's the daytime and nighttime asthma diary. And we spent a lot of time on digital outcomes, not just because it's new and fun, but because we believe there's real utility there. They're continuous and non-invasive. They're a great alternative to PROs because they require minimal input from the patient. They also have a wide dynamic range because they're truly continuous endpoint. And great examples of how this might work are cough counting and also sleep disturbance monitoring. And moving on to how we design phase 2 efficacy studies, before I was in the drug development space and when I was in academics, I remember reading papers and thinking, why did they do it that way? And the answer is quite complex, as you might guess, but it really depends on the objective of the study. And the problem with phase 2 studies is we have a very mixed set of objectives where one of our remits is to achieve proof of activity as soon as possible. But our other remit is to increase the likelihood of success in phase 3 if the program is going to move on. And these two objectives are frequently competing with each other. And I have an example in asthma to demonstrate how these objectives might conflict with each other. Inhaled allergen challenge is a model that's been used to show proof of biology and proof of activity in asthma. And for Th2 biology, it's a very relevant model. And frequently you can get an answer within a year, year and a half, with a small number of subjects on whether a molecule has activity in that disease model. However, it is a disease model and not a reflection of how asthma patients actually experience their symptoms or exacerbations. And so alternately, you may have seen in the literature and in the clinical trial space that many larger phase 2 studies in the asthma space mirror phase 3 as much as possible. And they do that in a few different ways, whether it's the primary endpoint, which is usually exacerbations, because that is the main way asthma therapies are being approved. And also payers look at that endpoint as an important part of where that brings value to their asthma patient. Also, these larger phase 2 studies try to mirror the phase 3 population as much as possible. And also quite importantly, since standard of care may vary by regionality and genetic susceptibility may vary by regionality, larger phase 2 studies try to mirror the regions that will be studied in phase 3 as well. And as I'll show at the end of the talk, it turns out that seasonality is also very important in asthma. And this is one of the ways we see if our study is reflecting true asthma natural history. So exacerbations, I have to admit that clinical trialists in the asthma space have a love-hate relationship with exacerbations. They're a way to anchor how we compare therapies to each other and to the standard of care, but they have quite a few limitations, especially in the early development space. So you'll hear bias, I have to admit, has been informed by data. Exacerbations are a measure of risk in asthma. It turns out that a history of exacerbations is the best predictor of future exacerbations. And that's been reliably shown in observational studies and interventional studies. They're an uncommon event, usually less than one event per patient year. And for most recent studies that enrich for patients that have exacerbated in the past, that annualized exacerbation rate is about 0.6 events per patient year. Rate reduction, and I think this reflects the bias that I just mentioned, it's really a categorical endpoint masquerading as a continuous endpoint. And what do I mean by that? Well, you either have an exacerbation or you don't. And unlike other endpoints in other fields where they may use composite endpoints to define their primary endpoint, exacerbations are only one way of looking at worsenings of asthma. And we turn whether a patient has a exacerbation or the number of exacerbations they have over 12 months into a rate reduction that's then a continuous endpoint. Now, that's all well and good for a large phase 3 study. However, in a small phase 2 study, a small number of exacerbations in a study arm can have a dramatic effect on ultimately what that study looks like. And really what this means in statistics is that question is, how precise is that exacerbation signal in phase 2? And I think we can all agree that precision is a good thing. In terms of that transition to phase 3, it really plays a big part of what are the chances of us repeating that result in phase 3. So I have just two examples of precedent data that illustrate this variable precision and how it's dependent on sample size. So this used to be an internal molecule, no longer is, but levocisumab, 10 years ago at this point, the phase 2 data were published. And note that the sample size per arm was anywhere from 50 to 100, depending on how you split up that population. And this was split by Periosteum, which is a downstream biomarker of IL-13 activity. Levocisumab is a monoclonal antibody targeted against the ligand IL-13. And what I have boxed in red here is the range or the confidence interval of the rate reduction demonstrated in all patients or the high Periosteum group. And what's notable here first is that the treatment effect appears to be clinically relevant and large. 43% rate reduction in all patients and 67% rate reduction in the high biomarker group. However, because of the sample size and the relatively low number of exacerbations, that treatment effect is not very precise in this study. In fact, the interval crosses 0 for both, which means that in phase 3, the truth could be anywhere in that interval. And that's worrisome. And in fact, in later slides, I'll show that precisely that's what happened. We believe there was an overestimate of the true effect in phase 2 because of this imprecision. And ultimately, that program did not progress to filing for approval after the phase 3 studies in asthma. Now, we do learn from what we've done in the past, and astegolimab is an anti-SD2 antibody. It was most recently studied in asthma in a large dose-ranging phase 2b study, and the top dose studied was 490 milligrams. In the time that I submitted these slides for review at UW, this manuscript has actually been accepted for publication at JACI, so you should be seeing that relatively soon. And figure 1 in that publication shows the asthma exacerbation rate reduction for the top dose group across relevant populations. And the overall population, that rate reduction was 43%. And that confidence interval now, because the sample size is larger, there are about 125 subjects per arm in this study, that rate reduction estimate is now more precise. The 95% confidence interval here does not cross 0. And in populations that would be relevant to either thinking about Th2 biology or in clinical practice, as defined by the eosinophil count in peripheral blood at baseline, whether that cut was 150 or 300, in that low EOS group, especially when that EOS threshold is 300, there appears to be an enrichment for activity. But I think the take-home point in these two slides is that the phase 2 estimate of asthma rate reduction is not precise, and that precision is really defined by two parameters: the sample size of the study and the control group rate of exacerbations. So another important theme for us in thinking about precision in phase 2 study design is dynamic range. And in fact, the concepts of precision and dynamic range are linked. We see this in exacerbations. Because they're a relatively uncommon event, they have a low dynamic range. And this ultimately pushes the limits of what we can do in early development in terms of detecting a signal in a precise way. And I have two examples, and these are actually real world examples and will come up at the end of the talk. Example number one, and this actually looks pretty good on paper, a baseline exacerbation rate, annualized rate of 0.6, a 50% relative reduction in asthma is considered a large treatment effect. And in absolute terms, that means going from a baseline rate of 0.6 to a treatment group of 0.3 events per patient year. And in a 75 subject per arm study, this absolute difference is somewhere around 20 events. However, if now our baseline exacerbation rate is lower at 0.2, that 50% relative reduction translates to a much smaller absolute reduction of 0.1 events per year. And in a 75 subject per arm study, this absolute difference is now only 7 events. And I think this illustrates what I mentioned earlier, that a couple exacerbations here or there in a small phase 2 study where there's a relatively low exacerbation rate in the control arm can swing the efficacy needle wildly in either direction. And so this leads to the problem statement for why we're working on a different endpoint for asthma phase 2 endpoint development. How do we apply the concept of an acute worsening in disease to an endpoint with a larger dynamic range in order to obtain a more precise efficacy result? And the answer is quite simple, but how we get there is quite complicated. We need more observed events. And one of the ways to do that is to have alternate definitions of acute worsenings. And one of these ways is to reintroduce an old concept, but now in a more quantified way, of acute worsenings that don't quite get to the threshold of delivering a dose of systemic corticosteroids. And this concept, CompEx, was published in the Lancet Respiratory Medicine journal back in 2017. And CompEx is abbreviation for a composite endpoint of diarrhea worsenings and exacerbations. And those diarrhea worsenings include peak flow, rescue beta agonist use, and symptoms. This group, in their definition, required at least a worsening in peak flow and then one of the other parameters in diarrhea. They also incorporated the concept of a threshold effect versus slope criteria for meeting that diarrhea worsening. And this really tried to capture the concept that some patients feel an acute worsening, might be a mini catastrophic event in terms of an acute event, or they may have a more slowly progressing or a subacute progression of symptoms. Now, the CompEx event, as defined in this publication, is really driven by peak flow, and that was by definition, and then by rescue beta agonist use, and then symptoms. Their analysis was anchored in the effect size of exacerbations. And so in many ways, it was a bit of a self-fulfilling prophecy in that there was going to be a correlation demonstrated because it was anchored in exacerbations. And they split 12 studies into 2 different data sets. The first 5 studies were used as a learning data set to refine their algorithm for CompEx. And then the following 7 studies were used as test trials to refine and validate that algorithm. Now, within my organization, we had large data sets, and I think this is one of the advantages of being at a place with large precedent data sets. We can actually do quite a bit of internal validation and even reverse translation with the data sets that are in-house. We had a large phase 2b study called COSTA with Quolizumab. That is an antibody bound E receptor, not IgE receptor E. And Millie was that phase 2 study that I showed with that publication from New England Journal, Levocisumab. And so we try to validate what we saw in this publication internally. And just a little bit more about what the algorithm actually is. So again, CompEx is a composite of either a severe exacerbation or a diarrhea worsening. And that diarrhea worsening, as highlighted by the green boxes here, had to have at least a worsening in peak flow and either a worsening in reliever beta agonist use or a worsening in symptoms. And so by checking off which one a patient had, the boxes in green would qualify as a diarrhea worsening, whereas the intersection as shown in gray would not qualify as a diarrhea worsening. How good was this in terms of its correlation with exacerbations? Ultimately, what the authors showed in their publication and we validated in our internal data set was that a similar number of CompEx events were observed at week 12 as compared with the number of exacerbations observed at week 52 in data sets. This means when we look across studies that went into this data set, there are 2 to 3 times more CompEx events at week 52 as compared with exacerbations. Now, the way CompEx is reported is different than exacerbations. Exacerbations are reported as a rate reduction. CompEx in this publication and by us is reported as a hazard ratio of time to first event. And so first, we wanted to look at how good was that correlation of hazard ratio to rate reduction. And so when we look at exacerbations, this is not necessarily CompEx events. On the left panel, and this is, again, the advantage of sharing data across sponsors. This is pulled from the Transcelerate database that sponsors have agreed to feed into. When we look at the correlation between exacerbation rate reduction and time to first exacerbation hazard ratio, it looks like hazard ratio and rate reduction correlate quite nicely. And then the authors in the CompEx publication correlated the CompEx hazard ratio at 3 months to the exacerbation hazard ratio at 12 months, and that correlation was also quite good. Now, what this suggests is that CompEx at 12 weeks of treatment may be able to predict the effect that we see with exacerbations at week 52. Now, when we looked at published data versus internal data, in terms of how that phase 2 result may translate to a phase 3 result, and also how that hazard ratio of CompEx correlates with the exacerbation rate reduction. Again, this is where we pulled in data that were available to us either published or through the Transcelerate database. We do see that there's, on the left panel here, that there is a good correlation between the CompEx hazard ratio at week 12 and exacerbation rate reduction. And this really was an internal validation of what we saw in that publication. And more importantly, when we looked at data that were available to us either internally or externally, the CompEx result in phase 2 seemed to be as good as, if not better predictor of the exacerbation rate reduction in phase 3. So let me just point you to the lower portion of the right panel with the Levocisumab data. We have the asthma exacerbation rate reduction result in these green triangles. And then the CompEx result is in the red circle. And as we look at the difference between Millie, which is the phase 2 study, versus the phase 3 results, which were the Levolta studies, it seemed that the CompEx result lined up better with the phase 3 asthma exacerbation rate reduction than the phase 2 result in asthma exacerbation, and that this is actually a hazard ratio on this right panel. And as we looked at that in a more quantitative way in a table, if we look at both the Costa Millie study that I mentioned earlier, this expanded dynamic range gives us more precision in determining that effect size. And as I mentioned in the previous slide, when we look at these two studies together, at the end of the treatment period, there were approximately twice as many CompEx events than exacerbation. And that gives us more dynamic range, and that gives us more precision. And as I showed you graphically in that last slide, when we look at the Periosteum high subpopulation in that phase 2 study, in the first column here, we have exacerbations at end of treatment. In the middle column, we have CompEx at week 12. And then in the right column, we have CompEx at the end of treatment. The week 12 CompEx result correlated quite nicely to the end of treatment CompEx result, and were in fact quite different than the exacerbation result in terms of hazard ratio. And that hazard ratio of 0.49 or 0.51 was more in line with the phase 3 asthma exacerbation result. It turns out that hazard ratio of 0.5, depending on how you look at the relationship, correlates to a rate reduction of approximately 40 to 50%. I failed to mention at the beginning of my talk that I'm happy to take questions in the middle of the talk. And maybe this is a good point to pause to see if there are any questions up to this point. I'm not monitoring the chat window. And so if you have any questions, just please go ahead and jump in by audio. Now, moving on, unpublished data also to astegolimab or anti-ST2 results, and that study was coined Zenyatta. Because that study used a different diary than what was used in the publications and internal validation sets, this used the ADSD, ANSD, and importantly, that diary does not record the number of uses of relief beta agonists per day. In fact, it uses a qualitative as opposed to a quantitative question for that portion. We had to change the CompEx algorithm to try to get as close as we could to the original CompEx definition. And ultimately, because we couldn't use a reliever use, we looked at two different alternate CompEx definitions, which either used a threshold symptom change or a slope symptom change. And then in the second alternate definition, either a mean symptom change and a mean symptom change in slope. So that was a threshold change and a slope change, or a threshold change in each category, or a slope change. So that's a little complicated. Quite honestly, we were just trying to see if there are different ways of looking at the same data that got us closer to a more CompEx-like result. It turns out that either definition looked okay, and in fact, either definition, when compared to the other, performed quite similarly. And I think that result speaks to the fact that this concept holds true regardless of how we look at it. And I think there's strength in those data. Now, if we look at the correlation between the exacerbation rate reduction here on the SPAR graph is in dark blue, and then we look at the ADSD alternate definition of CompEx, and this is at week 12, and this is in light blue, across the treatment arms, whether it was placebo, 70 milligrams, 210 milligrams, or 490 milligrams, there appeared to be quite good correlation between the exacerbation rate. Sorry, this is I apologize. This is not actually rate reduction. This is the exacerbation event rate. Um, but the event rate that we noted, whether it was exacerbations or CompEx, were actually quite similar. And what this meant in terms of event rate reduction or hazard ratio, it turns out that the result was quite similar for exacerbations and CompEx. Now, translating this a bit now more forward-looking into how would we incorporate CompEx into an asthma efficacy study? We'd love to be able to get to proof of activity as early as possible in phase 2A. And this might look like a 2-arm study with at least 70 subjects per arm, maybe up to 100. But it turns out that magical number in terms of what we need to see in a blinded way, in a number of total events in order to see a real treatment effect. And that's with 80% power and with a less stringent two-sided alpha of 0.2. If we wanted to see that hazard ratio of 0.55, we actually only need a total number of 51 events across both arms to see that treatment difference. And that's not that many events. And in fact, this is what we incorporated into an ongoing asthma phase 2 study, where in a global study, this was a 2-arm study. We're going to treat patients for 52 weeks to get a readout on exacerbations, but there could be an interim analysis on CompEx events as soon as we hit a total number of 51 events. Now, this study that we incorporated this calculation in was designed pre-COVID pandemic. And just like many talks these days, we can't escape the COVID topic. It's still very real. But I did want to share in the last 2 slides how we think behavioral changes during the COVID pandemic may alter the natural history of asthma. And this has a profound effect, not only on our clinical trial, but you may have seen its effect on your clinical practice already. So this seasonality of asthma exacerbations is a theme that's been repeated in different publications. And it's a topic that Dave Choi covered with you a couple of years ago in this setting. We see 2 peaks in asthma exacerbations. In the fall, we see a peak as kids return to school, and the rhinovirus peak is in early September. And in the spring, because of outdoor environmental exposures, we see another peak. And this is most evident in an eosinophil high population, which is a rough correlation with Th2 high disease. And this was published by one of my colleagues in biomarker development in my organization, Tracy Staten, and others in Jackie in 2017. If we look at the phase 3 data set from Levocisumab, and we split the population by a blood eosinophil count at baseline of 300, in that EOS low population, there does not appear to be much seasonality in the asthma exacerbation rate. That seasonality then on the right is pronounced in the eosinophil high population, with a peak in September and a peak in February in the northern hemisphere. And you see on the bottom row that the data for the subjects in the southern hemisphere were shifted by 6 months so that we could complete this analysis. As I mentioned a couple minutes ago, we have had some profound behavioral changes as a result of the COVID-19 pandemic. Now, in the fall of 2020, in the northern hemisphere, most children were at home, and there was a lot less exposure to HRV as a result. And could face coverings and hand sanitizing also reduce exposures? Most likely so. We're just beginning to understand the magnitude of that effect. And as observed in an ongoing asthma study that we're conducting, we did not see, and this is in a blinded fashion, a peak in exacerbations in the fall of 2020. And in fact, that lower exacerbation rate in CompEx events has been quite dramatic. in this table, if we look at the anticipated exacerbation rate and what we've observed only a 0.2 annualized exacerbation rate as compared to what we anticipated of 0.6. And that CompEx event count, by this point in the study, we're anticipating 31 events, and we have only observed 16 events to date. This is an important change, I believe, in the natural history of asthma, but for us, that change in dynamic range dramatically reduces our ability to see a real treatment effect. And we've been discussing internally, are we even observing what we would consider a pre-pandemic natural history of asthma? Meaning, is asthma really the same? And how do we interpret those data, given the fact that we think the natural history of asthma has changed? We don't know what's about to happen. As kids go back to school this fall, maybe we'll see some of that natural history return. We also don't know what the spring will look like in terms of whether mask use will continue outdoors and whether we'll see a peak in exacerbations in the fall. And we really don't know when the seasonality that we know of will return to a pre-pandemic state. And along with that, when will we see a return to a pre-pandemic state in natural history? This is a real something that we're struggling with in terms of asthma clinical trials. And as a result, because we think asthma natural history has changed, we made a difficult decision to pause enrollment in this ongoing phase 2 study until we really figure out what the natural history of asthma looks like, until we really figure out what we're measuring. That is my last content slide. And I presented the work of many individuals, some of whom are no longer in my organization. Chris Kabansky was the lead statistician. He's now at the Parker Institute. Verena Steffen and Xiaoying Yang have also done a lot of these analyses. On the clinical side, Dorothy Chung and Horace Ree continue a lot of this work. And in biomarker discovery and biomarker development, David Choi and Tracy Stevens contributed to this body of work. And that is my last slide. I'm happy to take any questions you may have.
So are you running any asthma trials right now?
Technically, we are, but they are actually on pause. So they're still open. We haven't closed them. So the patients that were enrolled up to this date, we don't feel it's ethical to stop treatment because the drugs being tested, we believe, are still safe. And so they'll continue to end of treatment, but we've actually paused enrollment. So no new patients are being enrolled into those studies.
Could the fall flare in asthma exacerbations in part not be fully due to infection, since your low eosinophil population didn't seem to have a significant fall flare?
Yeah.
And the high eosinophil did. Could it be potentially driven in part by dust mite?
It may be. I think we're still learning what component of asthma biology are driven by eosinophils or correlate with eosinophils, right? And so it may be that there's still a link between infection exposure to the immunologic response that we see. And there may be a seasonal variance in dust mite exposure or other indoor exposures that we might see in the fall. Those are a bit more difficult for us to measure in the controlled setting. But I think because we didn't see, for example, I wouldn't expect necessarily house dust mites to change in the pandemic state. I wonder what published data sets we'll see from 20 and 2021 to show that this fall peak goes away when kids weren't in school. Our real world data scientists have been looking at claims data, and one of the data sets available to them is in the UK. And they've seen in both COPD and asthma that there are quite a few lower um, claims for new prescriptions of systemic corticosteroids that correlate with a clinic visit within a week. And that was accentuated in the fall. So I think, taking these two data sets together, we may be able to answer some of those questions that you've mentioned.
Any other questions? By the way, the word of the day is exacerbation. Again, exacerbation.
Well, if there are no other questions, again, thanks for inviting me. I think this theme of endpoint development is really important for what we do. We shouldn't be satisfied with the state of the art as it is today. I think we need to keep evolving. And I think we need to keep an eye on the natural history of asthma as it evolves, during this pandemic and post-pandemic. Thank you very much, and have a great day.
Thank you.